Have you ever looked at the vast blue ocean and wondered what's happening beneath the waves? Oceans are the lifeblood of our planet. They are massive, powerful, and full of incredible creatures. They give us oxygen to breathe and help control the weather, but they are so big, it can be hard to understand them. What if we could shrink the ocean down? What if we could build a tiny version of it right here in our lab? That was the big idea behind our project at Tiny Forest Lab. We wanted to create a small, self-contained ocean in a glass tank and watch it for 100 days. It's an epic experiment to see life in action. Our mission was simple but ambitious. We aimed to build a miniature ecosystem, a little world that could look after itself. We wanted to see how plants and animals would interact. How would they find food and shelter? How would the environment change over time? This wasn't just about making a pretty fish tank. It was a scientific adventure. By watching our small ocean, we hoped to learn big lessons about the real ones. We wanted to understand the delicate balance that keeps marine life thriving. This project was our window into the secrets of the deep, a chance to get up close with nature's amazing engineering. The excitement in the lab was electric as we began. An experiment like this is full of unknowns, and that's what makes it so thrilling. Would our little world survive? What amazing dramas would unfold inside this glass box? Every day would bring new observations and new questions. We would become explorers in our own lab, charting the progress of our tiny sea. We knew we would see life, and maybe even death. We would see competition and cooperation. It was a chance to witness the fundamental rules of nature playing out right before our very eyes, all within the span of just 100 days. So, why 100 days? This time frame gave us a fantastic opportunity to see real, meaningful changes. It's long enough for plants to grow and for animal populations to adjust. It allows the water chemistry to settle into a natural rhythm. A few weeks would only give us a quick snapshot, but 100 days would let us watch a full story unfold. We would see the initial chaos of a new environment transform into a stable, functioning ecosystem. It was a commitment, a long-term watch. We were ready with our notebooks and cameras, prepared to document every single moment of this incredible journey. Creating an ocean, even a tiny one, is a careful process. You can't just add water and fish and hope for the best. Our first step was to get the foundation right. We started with a large, strong glass tank, about 200 liters in size. This would be the vessel for our entire world. We cleaned it thoroughly to make sure no harmful chemicals were present. Then came the most important ingredient, salt water. We didn't just use table salt. We used a special marine salt mix that contains all the essential minerals and elements found in the real ocean. We mixed it with purified water, checking the salt level carefully with a tool called a hydrometer. Once the salt water was prepared and circulating in the tank, we needed to create the landscape. The bottom of the ocean isn't flat. It has mountains, valleys, and caves. So, we added a thick layer of special sand, called live sand. This sand is amazing, because it's already filled with billions of helpful bacteria. These tiny organisms are the cleanup crew of the ocean. They break down waste and keep the water clean. After the sand, we carefully placed live rocks. These aren't just ordinary rocks. They are porous, and also covered in beneficial bacteria and other small life forms, providing natural hiding spots and filtration. With the sand and rocks in place, our mini ocean needed the right equipment to keep it healthy. We installed a powerful filter to help remove waste from the water, just like the ocean currents do. We also added a water heater to keep the temperature stable, around 25 degrees Celsius, which is perfect for most tropical marine life. A protein skimmer was also essential. This clever device pulls organic waste out of the water before it can break down and pollute the tank. It makes lots of tiny bubbles that trap the gunk, which we can then remove. It's a game changer for keeping the water crystal clear. The final piece of the puzzle was light. Plants and corals need light to photosynthesize, which is how they make their food. We installed a special LED lighting system designed for saltwater aquariums. We could program the lights to mimic a natural day and night cycle. The lights would slowly turn on in the morning, get brighter during the day, and then dim in the evening. This natural rhythm is crucial for reducing stress on the animals and promoting healthy plant growth. With the water, landscape, and technology in place, our stage was set. Our miniature world was ready for its first inhabitants. Now for the exciting part, adding life. We couldn't put everything in at once, An ecosystem needs to be built slowly, layer by layer. The very first inhabitants were the smallest ones. We added more beneficial bacteria to kickstart the nitrogen cycle, which is nature's way of recycling waste. Then came the cleanup crew. This team of invertebrates is vital for a healthy tank. We introduced a dozen Nasarius snails, which bury themselves in the sand and stir it up, looking for leftover food. We also added several emerald crabs, known for munching on nuisance algae, and a handful of scarlet hermit crabs to scavenge for scraps. After the cleanup crew had a week to settle in, we introduced our first fish. We chose species that are hardy and known to be peaceful. Our first residents were a pair of Oceanography clownfish, famous from the movies. They are bright orange and incredibly charismatic. We also added a royal gramma, a stunning little fish that is half bright purple and half vibrant yellow. It loves to hide in the rockwork, darting out to grab food. These first fish were carefully selected to ensure they would get along and not compete too much for space or food in our relatively small environment. Of course, an ocean isn't just about animals, the plants are just as important. We wanted to create a lush underwater garden. We added a few different types of macroalgae. Unlike the fuzzy nuisance algae, these are beautiful, complex seaweeds. We chose a vibrant green species called Chaetomorpha, which we kept in a special compartment to help filter the water. In the main display we planted some beautiful red grape called lerpa, which has little bubbles that look like bunches of grapes. These plants provide oxygen, use up nutrients that could otherwise fuel bad algae, and offer shelter for tiny creatures. Finally, to make our world truly feel like a reef, we introduced some corals. We started with soft corals, as they are generally more forgiving than their stony cousins. We added a colony of pulsing xenia, which have feathery polyps that rhythmically open and close. We also included some green star polyps, which form a vibrant fluorescent green mat over the rocks. These corals added another layer of life and color to our tank. With the fish, invertebrates, plants, and corals all in place, our cast was complete. The 100-day countdown could officially begin. Our tiny ocean was now truly alive. The first 2 weeks were a period of intense observation. Every morning the team would gather around the tank, notebooks in hand, eager to see what had changed overnight. In the first few days, most of the inhabitants were shy. The royal gramma spent nearly all its time in a small cave it had claimed in the rockwork. The clownfish stayed close to one corner of the tank, but slowly, as they realized this new world was safe, their confidence grew. By the end of the first week, the clownfish were exploring every inch of the tank, and the royal gramma was making bold dashes into the open water to snatch bits of food. One of the first surprises was the sand bed. The Nasarius snails were doing their job perfectly, but they had help. At night, when the main lights were off, we'd use a red flashlight to look into the tank. We discovered a whole world of nocturnal activity. Tiny white starfish, no bigger than a fingernail, had hitchhiked in on the live rock. They were crawling all over the glass and sand, cleaning up microscopic bits of waste. We also saw tiny crustaceans called amphipods and copepods scurrying between the rocks. These critters are an essential food source for many fish and corals, and their appearance was a great sign. By day 10, we witnessed our first interesting behavior. The two clownfish had started to show signs of pairing up. One, slightly larger than the other, became more assertive. It would chase the smaller one away from food occasionally and perform a strange little wiggle dance. This is classic clownfish behavior for establishing dominance and forming a pair bond. The larger fish was asserting itself as the female. It was fascinating to see this natural instinct play out so quickly. They hadn't claimed an anemone yet, but they had designated a smooth patch of rock as their home base, which was a start. The plants also began to change. The green star polyps, which were just a small patch when we added them, had started to spread. A thin, purple mat was growing from the base, and new polyps were popping up on the edges. The macroalgae in the filter compartment was growing noticeably thicker, a clear sign that it was successfully absorbing nutrients from the water. However, we also saw the first signs of something less welcome. A fine brown dusting of diatoms on the sand and glass. This is a common part of any new aquarium, a sign the ecosystem is still finding its balance. As we moved into week 3 and beyond, our tiny ocean truly came to life. The initial shyness of the animals was completely gone. The tank was now a hive of activity from dawn until dusk. The clownfish pair became the undisputed rulers of the upper water column, confidently swimming in the open. The royal grama maintained its territory in the rockwork but was much more visible, its purple and yellow body a flash of color against the dark rocks. The hermit crabs were constantly on the move, comically clambering over rocks and each other in their endless search for a meal. The tank felt less like a collection of individuals and more like a community. The ecosystem was maturing. The initial bloom of brown diatoms faded away, consumed by the snails and outcompeted by the growing macroalgae. The water quality remained stable and clear, a testament to the hard work of the bacteria, the cleanup crew, and our filtration equipment. We started to see new life that we hadn't even added. Tiny feather duster worms with their beautiful spiraled crowns began to appear on the live rock. These filter feeders had also arrived as hitchhikers, and their presence showed that the water was clean and full of microscopic food particles for them to eat. Around day 40, something amazing happened. The clownfish started showing interest in the pulsing Xenia coral. At first, they would just nudge it gently. Then, they began to rub against its soft, feathery stalks. While Xenia is not a natural host anemone, clownfish in aquariums will often adopt a soft coral as a substitute. By day 45, they were fully hosting in the coral. They would sleep nestled within its pulsing polyps at night and rarely strayed far from it during the day. It was a beautiful example of animals adapting to their environment, forming an unexpected partnership that benefited them both. The middle weeks were also a time of growth. The green star polyps had now formed a lush carpet over their entire rock, waving gently in the current. The red grape caulerpa had sent out runners, and new bunches were sprouting up in different parts of the tank. The whole environment felt more established and robust. We had to start pruning the macroalgae in the filter every week, removing a handful of the green chetamorpha. This was a fantastic sign. It meant we were physically removing nutrients from the system, helping to keep the water pristine for all the inhabitants of our bustling underwater city. No experiment is ever completely smooth, and our 100-day journey was no exception. Around day 60, we faced our first major challenge. We noticed a patch of slimy, dark red algae starting to grow on a corner of the sandbed. This was cyanobacteria, often called red slime algae. While not truly an alga, it can spread quickly and smother corals and other life if left unchecked. Its appearance was a warning sign. It told us that there was an imbalance in the system. Specifically, it usually points to an excess of nutrients, like phosphates, that it can use for food. Our first step was not to panic. We had to investigate the cause. We performed a full range of water tests. While most parameters were good, we found that our phosphate levels had crept up slightly. It wasn't a dangerous level, but it was enough to give the cyanobacteria a foothold. We suspected the cause was a combination of things. Perhaps we had been slightly overfeeding the fish, and some food was decaying in the rocks. We needed to act decisively but carefully to restore the balance without shocking the entire system. Ripping it out manually would just make it release spores and spread. Our solution was a multi-pronged attack. First, we immediately reduced the amount of food we were giving the fish. We made sure they ate everything within a minute, with no leftovers sinking to the bottom. Second, we used a small siphon tube to gently suck out as much of the visible cyanobacteria as we could during our next water change. This removed the bulk of the problem directly. Third, we increased the water flow in that corner of the tank by adjusting one of our power heads. Cyanobacteria doesn't like strong currents, so this made the area less hospitable for it to grow. The final part of our plan was to boost our natural nutrient control. We added a special filter media to our system that is designed to absorb phosphates directly from the water. This acted like a sponge, soaking up the excess nutrients that were feeding the slime algae. It took about a week, but our plan worked. The cyanobacteria began to recede. The increased flow and siphoning kept it at bay, and as the phosphate levels dropped, it starved and disappeared completely. It was a valuable lesson in ecosystem management. Problems are signals, and by understanding the signal, we could find a natural balance solution. The final 30 days of our experiment were perhaps the most rewarding. After overcoming the cyanobacteria challenge, the ecosystem seemed to hit a new level of stability. The water was exceptionally clear, and all the inhabitants were thriving. The clownfish were now inseparable from their Xenia coral home, displaying vibrant colors and robust health. The royal grama had become surprisingly bold, often coming right up to the glass to inspect us as we watched it. Even the shy emerald crab was seen more often, confidently munching on tiny bits of algae on the main rockwork. The tank had found its equilibrium. One of the most fascinating developments was observing the food web in action. The tiny copepods and amphipods that had appeared early on were now a booming population. We could see them swarming on the glass at night. This provided a constant natural food source for the fish. We often saw the royal grama pecking at the rocks, hunting for these tiny crustaceans. This meant the fish were not solely reliant on the food we provided. The ecosystem was starting to produce its own food, making it more self-sufficient. This is a crucial milestone for any closed ecosystem, and a sign of true success. The corals also showed remarkable progress. The green star polyps had completely covered their original rock and were now starting to spread onto the neighboring rock. The pulsing xenia, home to the clownfish, had grown so much that we had to trim it back slightly to prevent it from shading other areas of the tank. This growth was a visual indicator of the health of our system. Corals are sensitive creatures, and their flourishing told us that the water temperature, lighting, and chemistry were all just right. Our miniature ocean was not just surviving, it was thriving and growing. On day 100, the final day of our experiment, we took a moment to just sit and watch. The tank was a dynamic, living world. The snails were bulldozing the sand, the hermit crabs were squabbling over a shell, the fish were weaving through the rockwork, and the corals were pulsing and waving in the current. It was a beautiful, complex dance of life that we had helped create and nurture. The journey from an empty glass box to this bustling miniature reef was complete. We had successfully simulated a small piece of the ocean and kept it healthy for 100 days. The sense of accomplishment was immense. This 100-day journey was more than just a fun project, it was a powerful learning experience. The most important lesson we learned was about balance. Every single element in our tank had a role to play. The bacteria processed waste, the snails cleaned the sand, the crabs ate the algae, the plants filtered the water, and the fish added life and energy. When we had our cyanobacteria problem, it was because that balance had been tipped, even just slightly. It showed us that a healthy ecosystem is like a finely tuned machine. If one part is off, the whole system is affected. We also learned a huge amount about patience. You cannot rush nature. We had to add livestock slowly, allowing the system time to adjust to each new inhabitant. We had to wait for the microscopic bacteria to grow and establish the nitrogen cycle. When we faced the algae problem, the solution wasn't instant. It took time for the changes we made to take effect, and for the ecosystem to heal itself. This experiment was a constant reminder that good things take time, and that observing and waiting are just as important as taking action. Nature works on its own schedule, not ours. The project highlighted the incredible resilience of life. We saw how the clownfish adapted and adopted a coral when no anemone was available. We saw how tiny creatures, the hitchhikers on our live rock, survived the journey to our lab, and then blossomed into a thriving population that became a cornerstone of our food web. Life finds a way to fill every available niche. Even when we faced a problem like the cyanobacteria, the ecosystem, with a little help, was able to bounce back. It showed us that while ecosystems are delicate, they also have an amazing capacity to recover if given a chance. Finally, we learned that the smallest creatures are often the most important. It's easy to focus on the colorful fish, but our tank would have failed without the invisible bacteria, the humble snails, and the tiny copepods. They are the foundation of the entire system. They are the janitors, the recyclers, and the first link in the food chain. This is just as true in the real ocean, where plankton, bacteria, and small invertebrates form the base of the entire marine food web. Our tiny tank was a powerful lesson that every single creature, no matter how small or seemingly insignificant, has a vital part to play in the great story of life. Our 100-day experiment in a 200-liter tank was a tiny window into a colossal world, but the lessons it taught us about balance, patience, and the importance of every creature apply to the great oceans that cover over 70% of our planet. Our real oceans are just like our tank, but on an infinitely larger and more complex scale. They have their own cleanup crews, their own cycles, and their own delicate balance that has been perfected over millions of years. Every part, from the giant blue whale to the microscopic plankton, is interconnected and essential for the health of the whole system. When we see problems like pollution, plastic waste, and overfishing in the real world, it's like the cyanobacteria outbreak in our tank. It's a warning sign. It's a signal that the ocean's balance is being disrupted. In our tank, we could quickly identify the problem and fix it. In the vast ocean, it's much, much harder. The excess nutrients in our tank were a small problem. The billions of tons of pollutants and plastics we pour into the sea are a catastrophic one. Our little experiment showed how sensitive an ecosystem can be, which makes the scale of the threats facing our oceans truly terrifying. But our journey also gave us a powerful sense of hope. We saw how resilient life can be, and how an ecosystem can recover if you give it a chance. By reducing the pollutants, excess food, and helping the cleanup crew, we restored balance in our tank. We can do the same for our real oceans. By reducing pollution, protecting important habitats like coral reefs and mangroves, and fishing sustainably, we can help the oceans heal themselves. The same principles apply. We need to identify the problems, reduce the stressors, and then let nature's incredible power of recovery do its work. Our little glass box of water became a universe of life. It taught us that an ocean, big or small, is a precious and intricate treasure. Protecting our planet's oceans isn't just about saving whales or dolphins, it's about protecting the entire system that supports all life on Earth, including our own. It's about ensuring the invisible bacteria can still cycle nutrients and the tiny plankton can still produce oxygen. Our 100-day adventure is over, but the real work of understanding and protecting our magnificent blue planet has only just begun. Every one of us has a part to play in keeping our one true ocean healthy for generations to come.